Dear students, today our topic is cell wall. We will know about its structure, chemistry, formation and functions. First, we will know about what is cell wall. The cell wall is tough, usually flexible but sometimes fairly rigid layer that surrounds many protists and virtually all fungal and plant cells. It is located outside the cell membrane and provides these cells with structural support and protection, and also acts as a filtering mechanism. A major function of the cell wall is to act as a pressure vessel, preventing overexpansion when water enters the cell. It was first discovered by Robert Hooke in 1665 in a thin slice of cork, a wall characterized by being thin and formed while the cell is undergoing rapid growth and elongation. The cell wall surrounds the protoplast, which includes the plasma membrane and all that it encloses. This membrane is usually pressed tightly against the wall because of the pressure of fluids inside. Many mature plant cells, especially those that have stopped growing, have laid down a secondary wall between the primary wall and the cell membrane. Between the primary walls of adjacent cells in the middle lamella that cements the two cell walls together. Now we will see structure of the cell wall. The cell wall consists of three main regions: the middle lamella, the primary wall and the secondary wall. In each region, the cell wall has three major components: cellulose microfibrils, hemicelluloses, and a matrix or encrusting material, typically pectin in primary walls and lignin in secondary walls. To understand the structure of these wall layers and their interrelationships, it is necessary to remember that plant cells generally do not exist singly in nature. Instead, they are adjacent to many other cells, and this association of thousands of cells taken together forms an organ such as leaf. Each individual cells must adhere to others in a coherent way to ensure that the cells can act as a unified whole. This means that they must be interconnected with one another to permit the movement of biochemicals, that is photosynthesis, hormones, cell signaling agents, etc., and so on, and water. This adhesion is provided by means of middle lamella, the layer of the cell wall material between the two or more cells, a part of which is contributed by each of the individual cells. This layer is the outermost layer of the cell wall continuum, and in non-woody organ is pectin rich. In case of wood, the middle lamella is lignified. The next layer formed by the protoplast just interior to the middle lamella is the primary wall. This primary wall is characterized by largely random orientation of cellulose microfibrils, like thin threads wound round and round a balloon in no particular order, where any microfibril angle from 0 to 90 degrees related to the long axis of the cell may be present. In cells in wood, the primary wall is very thin and is generally indistinguishable from the middle lamella. For this reason, the term compound middle lamella is used to denote the primary cell wall of a cell. Even with transmission electron microscopy, the compound middle lamella often cannot be separated unequivocally into its component layers. The compound middle lamella in wood is almost invariably lignified. The remaining cell wall domain, found in virtually all cells in wood and in many cells in non-woody plants or other plant pots, is the secondary wall. The secondary wall is composed of three layers. As the protoplast lays down the cell wall layers, it progressively reduces the lumen. The first formed secondary wall layer is S1 layer, which is adjacent to the compound middle lamella, or technically the primary wall. This layer is thin, characterized by a large microfibril angle. That is to say, the cellulose microfibrils are laid down in helical fashion, and the angle between the mean microfibril direction and the long axis of the cell is large, that is up to 50 to 70 degrees. The next wall layer is arguably the most important in determining the properties of the cell, thus the wood properties at microscopic level. This layer formed interior to S1 layer is the S2 layer. This is the thickest secondary wall layer and it makes the greatest contribution to overall properties of the cell wall. It is characterized by lower lignin percentage and a low microfibril angle from 5 to 30 degrees. There is a strong but not fully understood relationship between the microfibril angle of the S2 layer of the wall and the wood properties at a microscopic level. Interior to the S2 layer is the S3 layer, a relatively thin wall layer. The microfibril angle of this layer is relatively high and similar to the S1, that is up to 70 degrees. This layer has the lowest percentage of lignin in any of the secondary wall layers. The explanation of this phenomena is related directly to the physiology of the living tree. In brief, for water to move up the plant, there must be a significant adhesion between the water molecules and the cell walls of the water conduits. Lignin is hydrophobic macromolecule, so it must be in low concentration in the S3 layer to permit adhesion of water to the cell wall and thus facilitate transpiration. Now we will know what are the wall components, that is chemistry of the cell wall. The main ingredients in the cell wall are polysaccharides, which are built from monosaccharides or simple sugars. 11 different monosaccharides are common in these polysaccharides, including glucose and galactose. Carbohydrates are good building blocks because they can produce a nearly infinite variety of structures. There are a variety of other components in the wall, including protein and lignin. Let us look at all these wall components in detail. First, cellulose. Cellulose is the most abundant chemical on the face of the earth. It is a glucan polymer of the glycopyranose units, which are linked together by beta-1,4-glucan bonds. The building blocks for the cellulose is actually cellobiose. Since the repeating unit in cellulose is a 2-sugar unit, the number of glucose units in a cellulose molecule is referred to as a degree of polymerization. Beta-1,4-glucan is made of as many as 25,000 individual glucose molecules. Every other molecule, called as residue, is upside down. Cellobiose, that is glucose disaccharide, is the basic building block. Cellulose readily forms hydrogen bonds with itself, that is intramolecular hydrogen bonds, and with other cellulose chains, that is intermolecular hydrogen bonds. A cellulose chain will form hydrogen bonds with about 30 other chains to yield a microfibril. This is somewhat analogous to the formation of a thick rope from thin fibers. Microfibrils are 5 to 20 nanometers wide and give the wall strength. They have a tensile strength equivalent to steel. Some regions of the microfibrils are highly crystalline, while others are amorphous. Now, second, hemicellulose. Hemicellulose is a polysaccharide composed of variety of sugars including xylose, arabinose, mannose. They are characterized by being soluble in strong alkali. They are linear, flat, with beta-1,4-glucan backbone and relatively short side chains. Two common types of hemicelluloses include xyloglucans and glucuronoarabinoxylans. Other less common ones include glucomanons, galactoglucomanons, and galactomanons. The main feature of this group is that they do not aggregate with themselves. In other words, they do not form microfibrils. However, they form hydrogen bonds with cellulose and hence they are called cross-linking glycans. There may be a fucose sugar at the end of the side chain, which may help keep the molecules planar by interacting with other regions of the chain. Hemicellulose is abundant in primary walls but is also found in secondary walls. Now, third, pectic polysaccharides. Pectic polysaccharides are polymers primarily of beta-1,4-galactouranons. They are branched or unbranched polysaccharides and are helical in shape. Divalent cations like magnesium, calcium also form cross links to join adjacent to polymers, creating a gel. These are extracted from the wall of the cell with hot water or dilute acid or calcium chelators like EDTA. They are easiest constituents to be removed from the wall. They form gels that are used in jelly making. Although most pectic polysaccharides are acids, Others are composed of neutral sugars including arabinones and galactones. The peptide polysaccharide serves a variety of functions including determining wall porosity, providing a charged wall surface for cell-cell adhesion, or in other words, gluing cells together, that is middle lamella, cell-cell recognition, pathogen recognition, and others. Now, proteins. Structural proteins. In addition to the carbohydrates, cell walls contain a variety of proteins. One type of cell wall proteins are called glycoproteins, containing carbohydrate side chains on certain amino acids. One common group of cell wall proteins are characterized by having an abundance of amino acid, that is hydroxyproline. Structural proteins are found in all layers of the plant cell, but they are most abundant in the primary wall layer. Like the cell wall carbohydrates, glycoproteins are hydrophilic and can form hydrogen bonds and salt bridges with cell wall polysaccharides. In addition to hydroxyproline, cell wall proteins are often high in the amino acids proline and lysine. The amino on the lysine provides positive charges along the peptide backbone. The positive charge residues can associate with negatively charged groups on a pectic acid. In addition to electrostatic interactions, hydrogen bonds also form between amino acid side chains and cell wall carbohydrates. Another type of structural cell wall protein called extensin can form covalent bonds with other extensin proteins, though the amino acid tyrosine. In extensin, the tyrosines are evenly spaced, and when they bond with tyrosine on another extensin molecule, they can wrap around other cell wall constituents, knitting the cell wall together. The amount of extensin changes with development. Cells that have thick, hard walls are often rich in extensin, that is sclerites and fibers. The amount of extensin produced is dependent on mechanical wounding, infection, and these responses are mediated by plant hormones. Cell walls also contain functional proteins. Enzymatic activities in the cell wall include oxidative enzymes, that is peroxidases, hydrolytic enzymes, that is pectinases, cellulases, expansins, that is the enzymes that catalyze cell wall creep activity. General functions of the cell wall enzymes including protection against pathogens, cell expansion, and cell wall maturation. Now, lignin. Lignin is a heterogeneous and highly cross-linked macromolecule that represents the second most abundant natural polymeric material on the earth. It is a polymer of phenolics, especially phenyl proponides. Lignin fills the spaces in the cell wall between the cellulose, hemicellulose, and pectin components, especially in tracheids, sclerites, and xylem. It is covalently linked to the hemicellulose and thereby cross-links different plant polysaccharides, conferring mechanical strength to the cell wall and by extension to the plant as a whole. There are three monoligninol monomers methoxylated to various degrees, that is p-coumaryl alcohol, coniferyl alcohol, and snapyl alcohol. These are incorporated into the lignin in the form of the phenyl proponides, that is parahydroxyphenyl, guacyl, and syringyl. Gymnosperms have high lignin that consists almost entirely of G with small quantities of H, that is guacyl and hydroxyproline. That of dicotyledonous angiosperm is more often than not of mixture of G and S with very little H, and monocotyledonous lignin is a mixture of all three. Highly lignified wood is durable and therefore a good raw material for many applications. It is also an excellent fuel since lignins yield more energy when burned than cellulose. Mechanical or high yield pulp used to make newsprints contain most of the lignin originally present in the wood. This lignin is responsible for the newsprint yellowing with age. Lignin must be removed from the pulp before high quality bleached paper can be manufactured from it. Water. The wall is largely hydrated and comprises of between 75 to 80% water. This is responsible for some of the wall properties. For example, hydrated walls have greater flexibility and extensibility than non-hydrated walls. Suberin, cutin, and wax. A variety of lipids are also associated with wall for strengthening and waterproofing. Now, cell wall formation. How this cell wall is formed? The cell wall is made during cell division when the cell plate is parted between the daughter cell nuclei. The cell plate forms a series of vesicles produced by Golgi apparatus. The vesicles migrate along the cytoskeleton and move to the cell equator. The vesicles coalesce and dump their contents. The membrane of the vesicle becomes the new cell membrane. The Golgi body synthesizes the non-cellulosic polysaccharides. At first, the Golgi vesicles contain mostly peptic polysaccharides that are used to build the middle lamella. As the wall is deposited, the other non-cellulosic polysaccharides are made in the Golgi apparatus and transported to the growing wall. Cellulose is made at the cell surface. The process is catalyzed by enzyme cellulose synthase made in by the ribosomes, that is rough endoplasmic reticulum, that occur in rosette complex in the membrane. The enzyme apparently has two catalytic sites that transfer 2 glucose at a time, that is cellobiose from UDP glucose to the growing cellulose chain. Sucrose may supply the glucose that binds to UDP. Wall protein is presumably incorporated into the wall in a similar fashion. Cell wall is made from the outsides inward. Thus, as the wall gets thicker, the lumen, that is the space within the wall, gets smaller. Exactly how wall components join together to form the wall once they are in place is not completely understood. Two methods seem likely. First, self-assembly. This means that wall components spontaneously aggregate. And second, enzymatic assembly. Various enzymatic reactions are designed for wall assembly. For example, one group of enzymes that stitches xylons together in the wall to form a long chains. Oxidases may catalyze additional cross-linking between wall components, and pectin methyl esterase may play an important role. Now, functions of the cell wall. The cell wall serves a variety of functions such as, number first, it prevents the cell membrane from bursting in a hypotonic medium, that is registers water pressure. Number two, controls the rate and direction of the cell growth and regulates cell volume. Number third, has a metabolic role, that is some of the proteins in the wall are enzymes for transport secretion. Number fourth, physical barrier to pathogens and water in superacid cells. However, remember that wall is very porous and allows the free passage of small molecules. Number five, recognition process. For example, the wall of roots of legumes is important in nitrogen fixing bacteria colonizing the root to form nodules, and pollen stigma interactions are mediated by wall chemistry. Number six, economic processes. Cell walls are important for products such as paper, wood, fiber, energy, shelter, and even roughage in our diet. Number seven, physiological and biochemical activities in the wall contribute to the cell-cell communication. And number eight, thick walled cells provide the mechanical support. I hope that you have got what is cell wall, and I think you have got it very, very well. Thank you once again.